Alright, in this video we're going to go through some revision notes for Biology 0610, Cambridge IGCSE, for Topic 3, Movement in and out of the cell. So to begin with, cell surface membrane or cell membrane is a structure that surrounds cytoplasm and plays a role in movement of materials in and out of the cell by either diffusion, osmosis, active transport, or phagocytosis. We're going to go through each one step by step. First is diffusion. Diffusion is the net movement of molecules from a region of high concentration to a region of low concentration, moving down the concentration gradient as a result of random movement of particles through a partially permeable membrane. A partially permeable membrane is a cell membrane that can restrict the free movement of molecules, meaning it allows some molecules to pass easily, but others with difficulty or not at all. So diffusion is the movement of particles or molecules from a high concentration to a low concentration. For example, we have a high concentration of molecules here. Let's say we have about more than 10 to 20 here, and they move to a low concentration. Movement of molecules from a high concentration to a low concentration, moving down the concentration gradient as a result of their random movement, like gaseous particles and liquid particles have random movements. Always through a partially permeable membrane. Partially permeable membrane. A membrane that's partially permeable. So it is permeable. Particles can pass through, but they can partially pass through. So it allows some particles to pass through, others may not. Why is diffusion important for living organisms? In our syllabus, these are the 3 points we need to remember. The first one, to obtain the requirements. The second one, to get rid of waste products. And the last one, carry out gas exchange for respiration. Example, plants need oxygen and carbon dioxide for respiration. This is a diagram showing diffusion. You have a high concentration of particles here, moving through a partially permeable membrane. As you can see, all the particles are not going through the membrane. Only some are being able to pass through to a low concentration. So high concentration to low concentration through partially permeable membrane. These are some examples for diffusion. The site, molecules moving from where to where. So in small intestine, the molecules moving are digested food products like glucose, amino acid, fatty acids, and glycerol. From the lumen of the small intestine to blood and lymph in the villi found covering small intestine walls. Villi are finger-like projections in the small intestine to absorb nutrients and minerals. Second site is the leaf. The molecules moving in the leaf is oxygen. Oxygen moves from air spaces between mesophyll cells to mitochondria in all cells. In the leaf also we have carbon dioxide, which moves from air spaces between mesophyll cells, usually spongy mesophyll, to chloroplasts in mesophyll cells, usually from spongy to pellucid mesophyll. Leaf again, molecules moving is water vapor from stomata to air outside the stomata. The pores in the stomata to air outside. Lungs transport oxygen from alveoli airspace to blood in capillaries around the alveoli. And lungs also transport carbon dioxide from blood in capillaries around the alveoli to alveolar space. Energy for diffusion. Actually, diffusion does not require any external source of energy, because the source of energy, or the energy for diffusion, comes from Brownian motion. Brownian motion is random movement of particles in liquids or gases due to their kinetic energy and bumping into one another. So Brownian motion only occurs in fluids. For sure, solid is not a fluid because its particles are not able to move around in random movement. Liquids and gases are fluid, so they have random movement of particles, and due to their bumping into one each other and their kinetic energy and vibration, this brings the energy for diffusion. What are some factors affecting diffusion? The first one is surface area to volume ratio, inversely proportional. So a big cell structure has a small surface area to volume ratio and slower diffusion. A small cell structure has a big surface area to volume ratio and faster diffusion. Examples for cells adapted for diffusion having a high surface area is root hair cell, found in plants, to absorb water and mineral ions, have large surface area and thin walls to absorb water. Ileum in humans, to absorb product of digestion and the small intestine. This is the root hair cell, and this is the ileum epithelial cell. The second factor affecting diffusion is distance, also inversely proportional. High distance is equal to slow diffusion, and small distance is equal to fast diffusion. So just imagine two doors next to each other, one door and the other door, a parallel distance between them, and this is one door and this is the other door. If you want to run from this door to this door, it's going to take you more time here, while to run from this door to this door is going to take you less time here. So the smaller the distance, the faster the diffusion. The smaller the distance, the faster the particles are transported from one source to the other, from a high concentration to low concentration. But the more the distance, the more time it takes for the particles to move from a high concentration to the low concentration. And this is why blood capillaries and alveoli have walls only one cell thick, to ensure rate of diffusion across them is as fast as possible. The third factor affecting diffusion is temperature, directly proportional. Low temperature is equal to slow diffusion, high temperature is equal to fast diffusion, because high temperature results in more collisions with the cell's membrane and Brownian motion, which increases the kinetic energy of the cell, allowing for more faster and efficient diffusion. The fourth factor affecting diffusion is concentration gradient, also directly proportional. High concentration gradient is equal to faster diffusion, and low concentration gradient is equal to slow diffusion. Alright. Solution. Solution is a mixture of solute dissolved in a solvent. Solute are the particles that dissolve in a solvent, and solvent is the fluid that dissolves a solute. So, for example, we have a beaker of water here, a beaker containing water, and we add particles of sugar inside. So the solvent is the water, because the solvent dissolves the solute. So this is the solvent. It dissolves what? It dissolves the sugar. So sugar is the solute. Solute is the substance that goes into the solvent, and solvent is the substance that dissolves the solute. For example, in living organisms, the solvent is water, the solute is glucose, and the solution is an aqueous solution. Aqueous solution are a solution containing hydrogen and hydroxide to combine and form water. Water as a solvent. Water is the most common solvent. And what is the importance of water for organisms? There are four importances of water for organisms. The first one, it can easily be transported around organisms. It helps move digested food molecules to cells all over the body. Part of the cytoplasm and plays important role in ensuring metabolic reactions or expo reaction. It dissolves urea and other excess substances as salts to remove from body in urine. This is an example of solute and solvent. Sodium chloride dissolved in water. Here, water is the solvent in the beaker. This is the solvent. And the particles of sodium chloride dissolved. This is the solute. There are two types of solutions, dilute solution and concentrated solution. Dilute solution is a solution with high water potential, meaning high concentration of water molecules and low concentration of solute particles. Concentrated solution is a solution with low water potential, meaning low concentration of water molecules and high concentration of solute particles. All right. Osmosis. Osmosis is, in short, the diffusion of water, meaning osmosis is the net movement of water molecules from a region of high water potential and concentration with less solute, which is a dilute solution, to a region of low water potential and concentration with high solute, which is a concentrated solution, through a partially permeable membrane. So during osmosis, water molecules, not particles, not any molecule, but water molecules in specific, move from a region of high water potential, so they move from a dilute solution to a region of low water potential, a concentrated solution. During osmosis, water moves down the water potential gradient, and water moves in and out of cells by osmosis only. This is why we call it the diffusion of water. Diffusion, high to low gradient of water moving down the water potential gradient from dilute to concentrated solution, high water potential to low water potential. This is an example of osmosis for glucose. There's two examples. Let's understand this one first, then the other one. On the left side, we have a sugar solution. This is concentrated. Concentrated means we have more solute particles, so more sugar and less water. The right side is sugar solution dilute. Dilute means less solute particles and more water particles. Now, during osmosis, if we return back to the definition, osmosis is a net movement of water molecules from a high concentration to a low concentration. So, in the right side, dilute solution, water will move from a dilute solution with high water potential to a concentrated solution with low water potential, moving down the concentration gradient. In the next example, we have a strong glucose solution and a weak glucose solution. This is the same example as this one, it's just a different way of drawing the diagram. Strong glucose solution, strong means concentrated, so a high number of glucose molecules. Weak glucose solution means less number of glucose molecules. This means dilute, this means concentrated. This is dilute. So we have a high number of water molecules, so high water potential. If this is concentrated, we have high number of solute particles and low number of water molecules, so low water potential. So water will move through partially permeable membrane from high water potential to low water potential down the water potential gradient. showing osmosis through a partially permeable membrane. Since the cell membrane is partially permeable, it allows small molecules as water to pass through, but not larger like solute. So this is like a partially permeable membrane. It allows the water molecules to pass through, but since solute molecules are a bit larger, it doesn't allow solute molecules to pass through. Now we're going to study osmosis in plant cells and osmosis in animal cells. First, osmosis in plant cells. We have two scenarios in osmosis in plant cells. The first scenario: if a plant cell is placed in a dilute solution with high water potential, example distilled water, what happens? Water moves into the plant cell via osmosis, causing the cytoplasm to swell up and making the vacuole bigger, till water molecules push the cell membrane against the cellulose cell wall, increasing the turgor pressure in the cell to make them rigid and turgid. So what's happening is you have a plant cell and you place it in distilled water, a dilute solution with high water potential. Now the solution you put it in has high water potential. If the solution has high water potential, it means the plant cell has low water potential. So water will move from high water potential in the solution to low water potential in the plant cell via osmosis. This movement of water into the cell will cause the cytoplasm of the plant cell to swell up and make the vacuole bigger. As the vacuole gets bigger, it will start pushing against the cell membrane. The cell membrane will push against the cellulose cell wall, and this will increase the pressure over all the cell, and it will make the cell rigid or turgid. What is the importance of scenario 1? 3 importances. The first one, it provides support and strength for the plant, making the plant stand upright with leaves held out to catch sunlight, of course for photosynthesis. The pressure created by cell wall stops too much water from entering and prevents the cell from bursting. If plants don't receive enough water, cells cannot remain firm and turgid, and they begin to wilt. This is a diagram of a turgid plant cell. As you can see, the vacuole is quite swollen up, and the cell wall is being pushed against the cell membrane. Now the second scenario in osmosis in plant cells. If a plant cell is placed in a concentrated solution with low water potential, what happens? Water moves out the plant cell via osmosis, causing cytoplasm and vacuole to shrink till cell membrane has pulled away from the cell wall, making it flaccid or plasmolized. So what's happening in scenario 2 is you place a plant cell in a concentrated solution with low water potential. If you put the plant cell in a solution with low water potential, this means the plant has high water potential. So water will move from high to low water potential from the plant cell to outside the plant cell in the solution. This means the plant cell will lose water into the solution. As the plant cell loses water via osmosis, the cytoplasm and vacuole begin to shrink, and the cell membrane becomes detached from the cell wall. This is known as flaccid or plasmolized plant cell. This scenario negatively affects the plant's ability to support itself since the cell wall has become detached from the cell membrane. See? It's detached. This means it's plasmolized or flaccid. Osmosis in animal cells. Animal cells lose or gain water as a result of osmosis. Since they have no cell wall, its results on the cell are more severe and damaging. For osmosis in animal cells, we use the 3 terms, so it's very important to remember these 3 terms. Hypertonic solution means a concentrated solution, meaning it has a low water potential and high solute molecules. Hypotonic solution is a dilute solution with high water potential and low solute molecules. Isotonic solution is a balanced solution. equal number of water and solute molecules. Now, what will happen if we put animal cells in each one of these solutions? If we put animal cells in a hypertonic solution or concentrated solution which has low water potential, so putting a animal cell in a solution with low water potential, hypertonic, okay, the animal cell has high water potential, so the cell will lose water by osmosis and it will become crenated or shriveled. Let's refer to the diagram. This is the animal cell. As a result of putting the animal cell in low water potential, the animal cell has high water potential, so it loses water and becomes crenated. Second is placing an animal cell in a hypertonic or dilute solution with high water potential. So if we put a animal cell in high water potential, this means the animal cell has low water potential, so the animal cell will gain water. Water will move inside the animal cell via osmosis, but since the plant cell does not have a cell wall to support it, it will increase the turgor pressure in the animal cell, and this will cause the animal cell to burst. This is known as hemolysis. The last one is putting an animal cell in a hypertonic solution. Nothing will happen if we put it in a hypertonic solution. It will remain the same because net movement of water in and out is equal. Now we have finished diffusion, osmosis in animal cells and osmosis in plant cells. Now we're gonna go through an osmosis experiment. The most common osmosis experiment, or at least the one that is included in our new syllabus for Biology 0610, 2024 to 2025, is cutting cylinders of root vegetable and placing them into distilled water and sucrose solutions of increasing concentration. To perform this experiment, the cylinders are first weighed before placing into the solutions. Then they are left in the solution for 20 to 30 minutes. Then they are removed. Then they are dried to remove excess liquid, and then they are re-weighed again. So first, you weigh them by placing them into solutions. Then leave them in the solution for 20 to 30 minutes. Then remove them again, dry them to remove the excess liquid, and then re-weigh them again. After performing the 5 steps, there are 3 possible cases that can occur. The first one, if plant tissue gains mass, this means water moved into the plant tissue from the solution by osmosis. So this means the solution is dilute with high water potential, and the plant tissue is concentrated with low water potential. If the plant tissue loses mass, water moved out of the plant tissue into the solution by osmosis. This means the solution is concentrated, low water potential, and the plant tissue is dilute, high water potential. If there is no overall change in mass, this means there was no net movement of water, and the concentration in both the tissue and the solution are the same. The next osmosis experiment is by dialysis tubing. Dialysis or whisking tubing, same thing, is a non-living partially permeable membrane made from cellulose. The pores in this membrane are small enough to prevent passage of large molecules like sucrose, but allow small molecules like glucose and water to pass through by diffusion and osmosis. The theory behind dialysis tubing demonstration. First, fill a section of dialysis tubing with concentrated sucrose solution. Then suspend the tubing in a boiling tube of water for a set period of time. Water will move from a region of high water potential dilute solution to a region of low water potential concentrated solution through a partially permeable membrane. This is the example. All right. This is a paper 6 note. Very important. Paper 6 note. This comes in all paper 6 and most paper 4 questions. Independent variable, dependent variable, and controlled variable. These are types of variables in an experiment, not just osmosis experiments, but in all experiments. And not just for biology, but in all experiments in science. Independent variable is the one thing that you can change in an experiment, maximum only one independent variable in an experiment. Dependent variable is a measurable or measured change that occurs because of an independent variable. Controlled variable is everything that remains constant or same during the experiment. For example, placing the same plant in different solutions. So this is the same plant, we place it first in a solution of cola, then in a solution of juice, and last in a solution of water. The independent variable is different solution because this is the one thing we change in the experiment. The dependent variable, or something we can measure, is the plant growth, height, length, and the number of leaves. The controlled or constant variables, the variables that remain the same throughout the experiment, we cannot change them. They are the same species, same age, same length, and the same solution of volume. Of course, there are many more, but I've mentioned only the most common ones. And with that, we have finished the first movement method in and out of cells through the cell membrane, diffusion. Now, the next method is active transport. Active transport is somewhat like the opposite of diffusion, because in active transport, we have the movement of particles through cell membrane from a region of a region of low concentration to a region of high concentration using energy from respiration. Energy from respiration is needed in active transport to move particles against the concentration gradient, in the opposite direction of which they would naturally move, like in diffusion. So in diffusion, the particles move from a high to low water potential in osmosis, diffusion, concentration gradient, through partially permeable membrane without using energy. They use Brownian motion. In active transport, it's a movement of particles through the cell membrane from low to high concentration gradient, moving in the opposite direction in the concentration gradient, using energy from respiration. Why does active transport need energy but diffusion does not need energy? Imagine you're walking on a rooftop. As you're walking on a rooftop, and suppose you suddenly fall off. What happens? You don't need energy to fall off. Gravity automatically pulls you down. There's no need for you to do additional work to fall down. But to go from the ground floor up to the rooftop of a building, you need energy to pull you up. So this is why active transport needs energy to make the particles move in the opposite direction they would naturally move. This is active transport in a cell. You have partially permeable membrane. It occurs by using carrier molecules. We'll learn about them in the next page. Concentration gradient, low to high. What is the importance of active transport? In our syllabus, we have two importances of active transport. The first one is uptake of glucose by epithelial cells in the villi of small intestine and the kidney tubules and nephrons. The uptake of ions from soil water by root hair cells in plants. Carrier proteins in active transport. Active transport works by using carrier proteins embedded in the cell membrane. How does it work? First, the substance combines with carrier protein molecule in the cell membrane. Then carrier transports substances across the membrane using energy from respiration to give them kinetic energy to change the shape and move the substances through the cell membrane. Then the substance is released into the cell. This is active transport. This is the carrier molecule. It takes in the substance, and the carrier molecule or protein takes it in, changes the shape, and sends it out from a low concentration to a high concentration, moving against the concentration gradient. And with that, we have finished all the methods of transportation except phagocytosis, because phagocytosis, we're going to talk in detail about phagocytosis in chapter 9, circulatory system, when we talk about white blood cells and how phagocytes perform phagocytosis. A summary of passive transport and active transport. Now, passive transport and active transport, these terms are not included in our syllabus, but this is just the general term to define diffusion, osmosis, and all. In passive transport, we have diffusion and osmosis. Diffusion is for all molecules, osmosis is only for water. Both move from high to low concentration gradient, no need from energy because they use Brownian motion. And both of them move through partially permeable membrane. The second one is active transport. Active transport, this is included in our syllabus, but active transport and passive transport, these two terms to differentiate are not included in the syllabus. And exocytosis and endocytosis are the two types of active transport, also not included in the syllabus. This is just for general information. Active transport depends on lower to higher concentration gradient, and it needs energy from respiration. This is a quick comparison between respiration and photosynthesis. Respiration is in both plants and animals. It occurs in the mitochondria. At all times, it takes in oxygen and releases carbon dioxide. Photosynthesis occurs in plants only. It occurs in the chloroplast, when chlorophyll captures energy from sunlight and uses water and carbon dioxide to convert it into glucose and oxygen. It happens only at the daytime when sunlight is available. It takes in carbon dioxide and releases oxygen.